There are 4 DNA bases. All scientists are familiar with these: adenine, thymine, guanine and cytosine, or ATGC. These base pair up, A with T, G with C, with 2 or 3 hydrogen bonds to give us a strand of DNA. Research published last week in Nature has demonstrated what scientists have suspected for a while. There aren't 4 DNA bases, so much as 4 natural DNA bases. In the study, scientists successfully modified an organism to contain unnatural base pairs, or UBPs. These unnatural base pairs are named 5,6 and NAM, and just like A and T or C and G, they pair up. The only difference is that rather than hydrogen bonds, hydrophobic interactions keep 5,6 and NAM together. This is the same phenomenon as when you see oil and water not mixing. The carbon rings are hydrophobic and would much rather interact with each other than the aqueous environment of the cell. Scientists took bacterium E. coli, left the chromosome intact, and instead worked on a plasmid, an accessory piece of DNA not required for cell function. Into the plasmid they put a single base pair of a 5,6 analogue and NAM. The 5,6 analogue is a chemical that looks and behaves like 5,6, sort of. It base pairs with NAM just like 5,6, but scientists can tell it apart from the real 5,6. A transporter for the new bases was added to the cells, and they were cultivated on a medium containing the precursors to 5,6 and NAM. The cells were left to grow, and the plasmids were extracted. Sure enough, each one now contained a real 5,6 NAM base pair, proving that the cell's DNA replication machinery is able to recognise these unnatural base pairs and, if supplied with the precursors, implement them into the genome. So why is everyone really excited about this? Well, one idea is this could expand the genetic code. 3 DNA bases are used to encode an amino acid, which then makes a protein. In the natural genetic code, there are multiple ways of encoding most amino acids. We call this code degenerate as, despite being theoretically able to encode 64 amino acids, it only encodes 21, and the stop instruction. If we expand the genetic code to include 5,6 and NAM, we suddenly have hundreds of new possibilities. Adding just 2 letters to the genetic alphabet brings the total possible amino acids up to 216. One idea is that we could use the new bases to program cells to encode synthetic amino acids. Where exactly this will take us is not quite clear, and it certainly won't be for a while, but it will definitely be exciting.